Hello, I am Nalli from Virginia Commonwealth University, and I am the first author of the study entitled Augmentation of Cyclic GMP PKG Pathway and Colonic Motility by Hydrogen Sulfide. The work is part of my PhD dissertation performed in Dr. Karnam Murthy's laboratory. I and my co-authors are excited to share with you our findings. In the gastrointestinal system, nitric oxide from enteric neurons stimulates soluble guanylyl cyclase activity in the muscle cell, which increases cyclic GMP levels and activates cyclic GMP-dependent protein kinase or PKG, causing muscle relaxation. Cyclic GMP is regulated by cyclic GMP hydrolyzing phosphodiesterase or PDE5 enzyme, which is stimulated by the nitric oxide PKG pathway. Hydrogen sulfide is now identified as an important signaling molecule involved in regulation of muscle relaxation. Our previous studies in gastrointestinal smooth muscle identified the specific expression of cystathionine gamma lyase or CSE enzyme. Unlike nitric oxide, hydrogen sulfide does not generate cyclic GMP levels but modulates its levels in the cytosol. In this study, we tested the hypothesis that hydrogen sulfide increases cyclic GMP levels by inhibiting PDE5 activity. In rabbit colon smooth muscle cells, the nitric oxide donor, S-nitrosoglutathione, abbreviated as GSNO, caused a concentration-dependent increase in cyclic GMP levels measured by radioimmunoassay. This effect was augmented by L-cysteine, a substrate of CSE, or by sodium hydrogen sulfide, a donor of hydrogen sulfide. Similarly, GSNO induced a concentration-dependent relaxation of colonic muscle cells, which was augmented by L-cysteine or sodium hydrogen sulfide. The effect of L-cysteine or sodium hydrogen sulfide on cyclic GMP levels and muscle relaxation was concentration-dependent. PKG phosphorylates inositol 1,4,5-trisphosphate receptor, or IP3R, at serine 1756, which is measured as a proxy for PKG activation. PKG activity in response to GSNO was also augmented by L-cysteine or sodium hydrogen sulfide, which was blocked by a PKG inhibitor, 8-bromo-cGMPs, suggesting that L-cysteine and sodium hydrogen sulfide converge on PKG to mediate IP3 receptor phosphorylation. L-cysteine and sodium hydrogen sulfide also inhibit GSNO-induced PDE5 activity measured by ion exchange chromatography. The involvement of endogenous hydrogen sulfide in PDE5 inhibition was examined using CSE-specific siRNA, which suppresses CSE expression. The inhibitory effect of L-cysteine, but not sodium hydrogen sulfide, was blocked in CSE siRNA transfected cells, suggesting that the effect of L-cysteine was mediated via activation of CSE. Rabbit colon smooth muscle homogenates incubated with L-cysteine showed a significant increase in hydrogen sulfide production. L-cysteine-induced hydrogen sulfide production was blocked by the CSE inhibitor DLPPG but augmented by GSNO. This effect was attenuated by PKG inhibitor 8-bromo-cGMPs and completely blocked by CSE inhibitor DLPPG. These results suggest that activation of PKG by GSNO stimulates CSE activity and generates hydrogen sulfide. GSNO-induced relaxation in muscle strips was partly attenuated in the presence of CSE inhibitor DLPPG. These results suggest that GSNO-induced relaxation was partly due to hydrogen sulfide production via activation of CSE. Previous studies by Dr. Greider's group have shown that release of calcitonin gene-related peptide, or CGRP, initiates the peristaltic reflex. Flat sheet preparation studies were performed with the help of our colleagues Dr. Greider and Dr. Kendig. L-cysteine did not affect the basal release of CGRP, however, it further increased CGRP release at 4 or 8 strokes of mucosa. This effect was attenuated by CSE inhibitor DLPPG, which suggests the contribution of hydrogen sulfide to CGRP release. L-cysteine also significantly increased the velocity of pellet propulsion in mice colonic segments, suggesting that hydrogen sulfide stimulates colonic transit. In conclusion, we demonstrate a unique interplay between nitric oxide and hydrogen sulfide in gastrointestinal smooth muscle. We show evidence that PKG activates cystathionine gamma lyase and generates hydrogen sulfide. Hydrogen sulfide in turn inhibits phosphodiesterase 5 activity and cyclic GMP hydrolysis, thereby augmenting cyclic GMP PKG pathway and muscle relaxation. We also show that hydrogen sulfide stimulates colonic transit. Restoration of nitric oxide and hydrogen sulfide homeostasis within the smooth muscle may provide novel therapeutic approach to restore motility disorders. Thank you for your attention to our study.